हेलो स्टूडेंट्स, मेरा नाम एलेक्स मैथ्यू है और मैं आपका बायोलॉजी एजुकेटर हूँ। आज का ये जो सेशन है इट विल हेल्प यू टू अंडरस्टैंड द कॉन्सेप्ट बिहाइंड द एनालिटिकल एंड द न्यूमेरिकल क्वेश्चंस दैट हैव बीन आस्क्ड इन नीट प्रीवियस ईयर क्वेश्चंस। सो जो लास्ट 10 years में जो मैंने कॉमन क्वेश्चंस देखे हैं, सो आई जस्ट मेड अ presentation of that, and we will be discussing the concept how to solve these questions in a quick and easy manner. ठीक है। सो आप अगर इसके पीछे के जो क्वेश्चंस जो हैं, इसके पीछे के कॉन्सेप्ट्स आप समझ गए, तो आप इजीली बाकी क्वेश्चंस सिमिलर टाइप के जो आपके नीट 2026 में आएंगे, इजीली आप सॉल्व कर पाएंगे। सो जो न्यूमेरिकल्स है या एनालिटिकल क्वेश्चंस है, वो मोस्टली इन टॉपिक्स में से रिपीटेड मैनर में नीट में पूछे जा रहे हैं, जो कि है जेनेटिक्स एंड प्रोबेबिलिटी, हार्डी वीनबर्ग, डीएनए रेप्लिकेशन और पॉपुलेशन इकोलॉजी। सो जो बायोलॉजी स्टूडेंट है, आई कैन बीइंग अ बायोलॉजी स्टूडेंट माई सेल्फ, आई हैव डन माई मास्टर्स इन बायोटेक्नोलॉजी और मैंने बहुत सारे बायोलॉजी रिलेटेड एग्जाम्स क्लियर करे हैं अपने लाइफ में। और मैंने मेरे करियर वाइज, आई हैव माय करियर इज इन टीचिंग। आई हैव more than 6+ years of experience of teaching biology to students. So हम लोगों को न्यूमेरिकल्स जब भी हम सुनते हैं तो हम डर जाते हैं। सो मोस्टली आई जस्ट वांट टू हेल्प यू इन टू अंडरस्टैंड दिस न्यूमेरिकल्स जो कि रिपीटेड मैनर में आ रहे हैं। और सिर्फ न्यूमेरिकल्स नहीं है इसमें, और एनालिटिकल क्वेश्चंस भी हैं which we'll be discussing. So the first question here we have regarding the Mendelian ratios. ठीक है। So let us try to read the question first. जो भी हमें क्वेश्चंस मिले उसको हमें बहुत धीरे से इजीली डिकोड करना है। हमें उसको डिकोड करना है, सिंपल एज दैट। सबसे पहला हमारा स्टेप यही है। सो फर्स्ट ऑफ ऑल वी विल ट्राई टू डिकोड दिस क्वेश्चन। So in a cross AaBb into AaBb, calculate the probability of offspring being. So genotypes दे रखे हैं यहाँ पे, एक होमोजाइगस डोमिनेंट है, एक होमोजाइगस रिसेसिव है, और यहाँ पे पूछा गया है कि एटलीस्ट एक डोमिनेंट एलील होना चाहिए। इसका मतलब ये है कि ऐसे जीनोटाइप्स कि हमारे पास एटलीस्ट एक होना चाहिए है डोमिनेंट एलील। नाउ एटलीस्ट का मतलब क्या होता है? एटलीस्ट मतलब एक, वन, और मोर देन वन, एक से ज्यादा तो होना चाहिए। इस क्वेश्चन का मतलब ये। नाउ वी हैव डिकोडेड द क्वेश्चन। नाउ लेट अस ट्राई टू सॉल्व इट। ठीक है। सो द पेरेंटल जीनोटाइप्स हमें दे रखे हैं। टू बीबी। ठीक है। नाउ वी नो दैट इसमें से वी विल गेट ए, वी विल गेट बीबी, here we will get AA genotype, we will get BB. ठीक है। This is what we know. Now when we cross this with this and this with this, now what is happening is AA genotype we are crossing with AA. Now the final phenotypes that will genotypes that will get is this. ठीक है। Now I hope that आपको ये हमें यहाँ पर ये कैसे मिल रहे हैं, we you all know about independent assortment, right? और उसके बाद here we are crossing BB into BB. ठीक है। So first we will get BB, then we will here we get BB, and again this, and finally this. Now if we go back to the question here, we need to calculate, हमें calculate क्या करना है? Homozygous dominant. ठीक है। हमें यहाँ पर ये मिल चुका है। Now अगर मेरे को homozygous dominant चाहिए है, तो मेरे को ए ए चाहिए होगा। तो ए ए यहाँ पे वन आ रहा है, कितने में से? फोर में से, वन आउट ऑफ फोर। और मेरे को सेकंड बीबी चाहिए, यहाँ मेरे को बीबी मिला है आउट ऑफ वन टू थ्री फोर, राइट। वन इंटू फोर इज 1:16। सो 1:16 will be the probability of genotype homozygous dominant genotype। ठीक है। Now if we go back to the question, the second part it is asking homozygous recessive. की probability कितनी होगी. Now AaBb, it is the second homozygous recessive, second part of the question that we need to find out. ठीक है. अब यहाँ पर जब हमने कैलकुलेट करा था, we got this genotype, we got this one, we got this and this. So only one homozygous recessive we got. So 1 out of 4 we got here AA. Now BB how much is there? It is 1 by 4 here also. So 1 by 16. The answer it is 1 by 16 of this. Then the final one, it is at least one dominant allele. मतलब एक dominant allele तो होना चाहिए. So यहाँ पर ये case possible नहीं है because this is homozygous recessive. So what we can do is we can minus one because probability एक ही होगी ना, उससे ज़्यादा नहीं हो सकती. We know that the maximum probability that can happen it is one. It cannot be more than one. We can then minus the homozygous recessive probability here. So this is the third part of the question. So here we get the probability of this, it is 1 by 16. So 16, 1, 16, here we solve this and we get 15 by 16 as the answer of the third one. So I hope that this question is clear, and if similar questions are coming, then you can solve in this manner. So now the next question we are having is from the Hardy-Weinberg equilibrium. यहाँ से question it can come in your NEET 2026, 100% I'm sure. So total number of people are 10,000. 900 individuals show the recessive phenotype. So here it is mentioned AA, so we can write here Q square is equals to 900. Now why I have written here as Q square? Now because we know that P it is the dominant allele and Q it is the recessive allele, right? So the formula we have P plus Q is equals to 1. Okay. And we need to calculate the allelic frequency P, Q and number of heterozygotes. So number of heterozygotes formula is 2 by 2 into P into Q. So first we need to find P and Q, then only we can get the number of heterozygotes. And we need to calculate the allelic frequencies, right? So now the frequency of the recessive phenotype AA here, it will be 900 divided by 10,000. So we will get 0.09. And if we need to get, this is Q square. If we need to get Q, we just need to take the under root, we will get 0.3. Now putting in the formula P by Q is equals to 1, we get P as 0.7, right? Now we just need in order to get the number of heterozygotes, we need to multiply 0.7 into 0.3, which we will be getting as 0.42. Now we need to calculate the number of heterozygotes, not the frequency, allelic frequency. It was mentioned as allelic frequency, that is why we had divided with the total number of population. Now, in order to get the number of heterozygotes, we need to multiply 0.42 into the total number of people, that is 10,000. So here we can calculate this as after 0 there are two digits, so 42 into 100 by 10,000. So 2 we can, two zeros we can cut here and we get the answer as 4200. So you need to remember two basic formulas, that is P plus Q, P plus Q is equals to 1, and the second formula it is the number of heterozygotes we get by 2 into P into Q. So now let us move to the next question. So this is a very important question. It has been asked in many of the NEET exams earlier also. So here we have the Meselson-Stahl um, Meselson-Stahl experiment, and the question it is related to it. So let us go to the question now. A bacterium with 100% heavy nitrogen labeled DNA undergoes 3 generations of replication in the N14 medium. So this is the light isotope of nitrogen. This is a light isotope, and this is the heavy. So find the total DNA molecules and the percentage of hybrid DNA molecules. So first of all, we need to find the total DNA molecules, and then we need to find the number of hybrid DNA molecules, right? So now how to proceed with this question? So here it is written that the bacteria is going through 3 generations of replication. So 3 generations of replication. So now let us take this as the DNA of the organism. So this is the 0th generation. So this is the normal manner in which the bacteria it is present. Now going, it is now going to double itself for the first time. So now we get the G1 generation. So here we know that it is replicating in a semi-conservative manner. So here we both have the heavy strands, right? This green color, it is representing the heavy strands because it is being labeled with heavy isotope of nitrogen, N50. Now it is being cultured in the light isotope, that is N14. Now when it is being cultured in the N14, now we know that DNA it is replicating semi-conservatively. So from the here we are getting the 2 strands. That is DNA, we know that it is double-stranded. So each one will have a light strand. Now they have become the hybrid, right? They have the heavy strand also with them, and they have the light strand also with them. So they have the light strand and they have the heavy strand with them. So first generation here, it is clear to us right now. Now again, they are grown on the light isotope, and then here we know that they replicate semi-conservative. So here we have one heavy strand. Again, we have one heavy strand. Then we have got one light strand and one light strand. From here we are getting right. Now this heavy strand it will accompany a light strand. We know that, and this light strand it will accompany a light strand itself. So this is the second generation. Now we are going to the third generation. So now we have one heavy strand, second heavy strand, and then we have one light, one light strand, one light strand, one here, and this. So a light strand will have a light strand itself, and a heavy strand will have a light strand. So now this is the third generation that we are getting. So the question is asking the total DNA that is present in the final generation and the number of hybrid molecules. So the number of hybrid molecules, it is only 2, right? Here we have the heavy, the green one, it is representing the heavy strand. The red one, it is representing the light strand. So we have 2 hybrids, and here we are having 1, 2, 3, 4, 5, 6, right? We are having 6 light strands, and total DNA we have, total DNA molecule it is 8, and we need to calculate number of hybrid molecules. It is the percentages. They have asked the percentage. So 2 divided by the total into 100, 1 by 4, 2 again. So it is 25%. So the answer it is 25% for the second portion. For the first portion, we are getting total DNA as 8. So I hope that you have understood this concept here. Now we can see that in each generation there are only 2 hybrids, right? There are only 2 hybrids in each generation. So this is a very important thing that I'm discussing right now, that in any generation, if you are taking after the G0 generation, any generation you are taking, the hybrid number of DNA, it will be only 2. If there is 3, 4, 10 generations, still the hybrid number, it will be 2. And the total number of DNA, it will be given by the formula 2 raised to n. So here it was asking how many generations? 3 generations. So n is the number of generations. So 2 is 2, 2 into 2, we are getting 8. So total DNA will be 8. So you can calculate in this manner also. But what if in the question they are asking how many light strands are there? Then you need to know that it will be there will be 6. So that is why I explained in this manner, so that question can be twisted in any manner. You know the basic and you know the trick also. So now here there were only 2 light strands. Here it has become, in the third generation, it has become 6. So if we go to G4 generation, the hybrid strand, it will remain the same, but the light strands, it will increase. So the next question, it is from population ecology, that is from growth rate. So the population grows from 500. So initial population, it was 500. The final population, it is 2000. The time it is given 2 years. So calculate the intrinsic rate of natural increase using exponential growth model. So we need to know the formula for this. So the formula it is, so this we need to calculate R. So formula it is ln nt by n0, that is divided by t. Now we know that nt it is 2000. Initial it is, we get 4. So what is ln4? So most people will get confused here. So you need to remember um, some important quantity. You need to remember it. So ln2 it is 0.693. So now here it is mentioned ln4. So it is 2 square. I can write like this. So whenever it is in this form, ln2 raised to 2. So it can be also written as 2 into ln 2. So 2 into ln2, kindly remember this is very important. It is 0.693. You need to remember some values. So here I remembered the ln2 values. So it is 0.693. So I get here, if I multiply with 0.693 into 2, the value that I will get, it is 1.386. Okay. Now I will put this in this equation, 1.3x divided by 2. So the answer I am getting is 0.693. So the R, that is the intrinsic rate of increase, it is 0.693. So kindly remember ln2, it is 0.693. So if you are getting ln4, you can just square it and do it in this manner. Ln 2 square, 2 into ln 2. Okay, ln 2 formal, ln2 value we know, so you just need to multiply 0.69. You can also remember ln4, ln8. You can just remember certain values which will help you to solve these type of questions. So the next question, it is from genetics. So the question here is that a color blind